Today we are here to discuss the behavior of the silver gull when present in differing gradients of human population density. In particular, we focused on the disturbance tolerance of the gulls before and after exposure to an anthropogenic intervention. As we are sure many of you are aware, the silver gull is a particularly bold and sometimes aggressive species that thrives in urban environments.
Indifference to human interruption is actually a pivotal evolutionary adaptation that is developed in many successful urban-dwelling birds, called disturbance tolerance.
Research conducted by Belland in 1997 and Smith in 1992 found that the success of the silver gull in non-native urban environments was largely due to this ability to adapt to the presence of invasive species and humans. Due to increasing habitat fragmentation and an increase in coexistence and cohabitation between humans and animals, a species that is hypersensitive to external stimuli and with a large flight initiation distance would be unable to survive in modern society. Therefore, this ability increases their survival rate and reduces undue stress. Alongside this, work done by Smith and Carr in 1993 suggested that the rise in silver gull population correlates with increasing human populations due to available food refuse. Taking all of this into consideration, we set out to design a study that aimed to investigate how anthropogenic intrusion affects the behavior response of the silver gull. We hypothesized that gulls in locations of higher human population density will be less affected by anthropogenic disturbance due to their acclimation to people being present.
The magnitude of this effect will be calculated by the activity budget pre- and post-disturbance of certain defined behaviours, as well as the time taken to resume normal pre-disturbance behaviour. In this case, normal behaviour includes preening, resting and foraging, and disturbed behaviour consists of startled departures, walking away from the subject, and increased vigilance.
We believe that our study will attempt to answer Tim Burgeon's questions in the following way. In regards to proximate mechanisms and immediate causation, we believe that the anthropogenic disturbance will act as a stimulus and the response that is elicited from silver gulls will be the observed change in behavior. For ultimate mechanisms, looking at function and adaptation, we believe the varying degrees of disturbance tolerance observed between flocks of silver gulls in the different locations will be a function of their evolutionary adaptations to increase their chances of survival. The silver gull's behavior would have adapted differently depending on the environment in which they need to survive and reproduce within.
Now that our observable behaviors are defined, the way in which they are affected by human disturbance can be assessed. Human disturbance in this case will be a subject walking into a flock of gulls and taking a seat. Behavior will be monitored by commencing recording 5 minutes prior to the subject entering and continuing recording 5 minutes after the interruption. We positioned or recording equipment 50 meters from the gulls and subject so as to remove any unwanted additional disturbance.
In order to evaluate if this behaviour is affected by what type of environment the gulls are accustomed to, 3 sites were selected to represent high, moderate and low population densities. Tumbalong Park grassland in Darling Harbour is a busy cityscape representing high density population. Bondi Beach in Sydney's eastern suburbs represented a moderate population density, while Camp Cove National Park is relatively isolated and provided a low population density. All recordings were performed at 11 a.m. during weekday mornings to reduce confounding variables such as temporal fluctuations of crowds during the day. Each site had observations repeated 3 times to give greater statistical power to our results, and each flock observed had between 10 and 20 gulls present in order to keep flock behaviour consistent. The same ethogram was applied to all site observations, and a combination of scan sampling of the entire flock at 15-second intervals and focal sampling of individual gulls allowed us to yield the following results.
The scan sampling here was divided between pre- and post-disturbance behaviour so that the activity budget for each could be compared. In Darling Harbour, we observed that once the anthropogenic disturbance occurred, resting behaviour was reduced. However, vigilance remained constant and preening surprisingly increased. The results from Bondi show that resting only marginally decreased, however preening stopped and foraging and walking remained constant. There was, however, an increase in startled flight response. Finally, the National Park observation showed that vigilance, along with taking flight response, increased in frequency, while resting behaviour decreased after the interruption. The focal sampling allowed us to measure the time taken to resume pre-disturbance behaviour. At each site, over the different recordings, 6 individual gulls were focused on. Here you see a graph of the average time taken to resume preening, resting and foraging once the vigilant or startled post-disturbance behaviour has ceased. While the high and moderate population densities are relatively similar, it can be observed that the low population density of the national park gave the longest time to return to normal behaviour.
While some of our results are inconclusive or even not what we expected, it is evident that the lower population density location yielded the highest proportion of startled responses and changes in alert and vigilant behaviour. This location also experienced the least amount of rest behaviour. These site recordings also show the quiet national park gull populations took the longest time to resume normal behaviour when disturbed.
Our experiment may have benefited from a greater sample size and a variety of different locations. Also, we must take into consideration the migratory ability of these birds, that they will experience many different locations of fluctuating anthropogenic disturbance, and the fact that they have many other confounding factors affecting their behaviour we could not control for.
This investigation gave some varied results, but our hypothesis was supported by the higher incidence of vigilant behaviour coupled with lower frequency of resting behaviour in the more isolated location. Their apparent lack of response to the stimuli, anthropogenic disturbance, is evidence of their adaptation to human intrusion and proof of their confident position within the shy-bold continuum.
Thanks for watching!